Welcome back, life science learners, to our second installment for today. We've looked at the process of what chromosomal mutations are. A reminder that we are looking at mutations today. In this segment, we're going to focus on what a gene mutation is. We're going to try and understand how this occurs, what are the factors that cause these mutations, and we're going to look at the possible effects of these mutations. So it's important that we recognize that gene mutations occur at a smaller scale when compared to a chromosomal mutation. This means that a gene mutation could affect one gene or a few genes. In comparison, when we looked at a chromosome, it meant that a chromosome has several genes on it, so the effect of that would be significantly more. What's important for us to recognize is that a gene mutation is a change in the sequence of the nucleotides in a gene. So if we go back to understanding what a gene is, it's basically a sequence of DNA nucleotides. So when a nucleotide or a base is added or is missing or changed, we say that that is a gene mutation. So it's important that we recognize that when we look at genes, a gene codes for a protein, and that happens through the mRNA being transcribed and then translated into a protein. However, when there is a change in the gene sequence, we often end up with an mRNA that has an abnormality or an altered sequence. And that will then translate into what we refer to either an abnormal protein or a protein that is not produced. And so we see now the effect of a mutation where the protein sequence has been altered. It's important that we recognize that there are certain factors that cause mutations, and we refer to those factors as mutagens. So any factor that causes a mutation is referred to as a mutagen. And so there are several different types of factors that cause mutations. So mutations can occur spontaneously, which means very unpredictably they can suddenly develop, and that's often because of the rapid rate of replication. We also know that there are certain environmental factors that can trigger these changes in the DNA. We know that there are certain ultraviolet lights that can be linked to the change in the sequence of nucleotides. X-rays and UV radiation can also result in changes in DNA sequences. We also find that there are chemicals, example benzene, formaldehyde, and carbon tetrachloride, that are all linked to be known as mutagens or mutation-causing agents. So we refer to those agents collectively as mutagens. So guys, a very good infographic on the various possible types of radiation or mutations we have. We know that we often tend to sunbathe or bask in the sun, and that sun radiation produces certain harmful ultraviolet radiations that can cause certain changes in our skin cells, and that can alter the sequence of cells, leading to cells dividing and growing uncontrollably, and we refer to that as cancer. We know that certain X-ray machines produce large number of X-rays that can obviously penetrate deep into our cells, especially our cells that are actively dividing, and alter the genetic sequence of cells in there. Smoking cigarettes contains lots of carcinogens or mutagen-causing chemicals, and these can alter our cells. We know that there are certain types of foods that contain carcinogenic or mutagenic substances. Certain products, as we said, chemicals can also be linked to mutations. Certain viruses and even bacteria can be linked to mutation or mutation-causing agents. One example of that is the HPV virus, which is linked to the initiation of mutations in your cervical cells. And so there's a concern around HPV infections and the link that has to in the initiation of cancer in certain cells. So guys, let's try and unpack what gene mutations are. So when we unpack what gene mutations are, it's important that we recognize that there are different types of gene mutations. And I'll broadly classify them into two groups, a point mutation and a frameshift mutation. At this point, let's try and understand the difference between the two. When we refer to a point mutation, it refers to a change at a single point, and that could include a nucleotide that has been altered, removed, or substituted. However, we also know that there are frameshift mutations, which then could change the length of the gene. So let's try and differentiate between the two and see the examples of these and the effects that they have. So a point mutation refers to a change in a single nucleotide in a gene. This may include a type called substitution. And so guys, the word substitution refers to a change. So think of when you're at school and you've got a substitute teacher coming into your class, it means there's a different teacher coming in, not the usual. So sometimes during DNA replication, we have accidentally a substitute nucleotide put into that sequence. And that changes the meaning, it changes the sequence present. And so we refer to that as a substitution. So this is a gene mutation where one nitrogenous base is replaced, again substituted, by another. And this may result, and the word may result, in an alteration of just one amino acid. And hence we refer to that as a point mutation. So if we try and unpack an example of that point mutation, so what I've illustrated in this table is a sequence of DNA as you would look up here. That's a sequence of DNA which I'm outlining in red. And so that sequence of DNA during the process of protein synthesis produces an mRNA codon or mRNA sequence. That mRNA sequence in turn is then translated into an amino acid chain. So when we look at this DNA sequence, produces a chain of methionine, lysine, leucine, alanine, and cysteine. However, if we subject or if we notice that there has been a point mutation, this would mean that on the mutated DNA strand that there would be a single nucleotide that has been affected. In the case of substitution, which is this example, we see that there has been a change or a substitute in the adenine nucleotide at this specific gene. And so we see that the G nucleotide has been substituted for an adenine. So here is a substitution that has occurred. So what does this mean now when we look at the mRNA sequence? It means that that DNA, when it's transcribed, now produces an mRNA that reads GUC. If we compare that to the original mRNA, which was a GCC, it means that now the amino acid in turn will be changed. So we had the original amino acid at that specific position called alanine. With the effect of substitution, where a single point mutation has occurred, we see that the amino acid at that specific point is now valine. And so in this case, the effect of a mutation has resulted in the amino acid being altered because of a single nucleotide change. And so we'll discuss the effects of that in a little while. We also know that there's a different type of point mutation called an inversion. So what does an inversion mean? An inversion essentially is a mutation where one or more bases in a triplet are inverted. So the word invert is to swap around. So I'll point that out in an example. This may result in the alteration of one or more amino acids. Let's try and understand that by using an example. So again, here's the sequence that we're looking at, and here's the inversion of that sequence. So essentially, the GAA has been inverted, and you see that it's now an AGG. What does this mean to the mRNA? When we look at the original mRNA, GAA coded for CUU. However, with the effect of the inversion, AAG now codes for a codon called UUC. What is the impact that it has now on the amino acid? The original amino acid for that specific codon was leucine. We now see that it has changed to an amino acid called phenylalanine. Again, the result of this means that there's been a change not just in the DNA sequence, but also a change in the amino acid sequence. What does this mean to the protein? This is a change in the structure of the protein. We know that proteins are defined by the specific sequence of amino acids. What will this mean to the protein? We'll have to unpack that when we look at the effects of these mutations. When we look at the other group of mutations, we refer to them as frameshift mutations. Just to recap, we're discussing point mutations, and in point mutations we said that there were different types, and we looked at a single base being inserted or substituted. Now we're looking at the next group, which is frameshift. So let's try and understand the different types of frameshift mutations. Again, the word frameshift refers to changing the length of the line. Now, a simple example of this would be, think of when you type in a specific sentence on your phone when you're texting someone. And often we make a mistake and we kind of add in an extra letter. And so that changes the length of your sentence. Or if we delete a word, it decreases the length of a sentence. So the impact that the frameshift mutation has is that it either changes the length of the gene, either by increasing it or decreases the length. And that can happen through either a deletion or, as I'm going to mention in the next slide, an insertion. Let's go back to deletion. So what does deletion mean? Deletion essentially refers to the omission, the removal. So as you would get the delete button on your keyboards, it means removing something. Sometimes a mutation results in a piece of DNA losing a nucleotide. Now, as I mentioned, let's try and identify the impact that it has on the original DNA. So here's the original DNA. It has a sequence of TAC, and that will transcribe into AUG. That in turn means that if a mutation took place, and let's look at what the mutation was here. Here we're seeing that the mutation took place where C was deleted. So this means that we now have a shift in the thymine from the next set of codons onto the DNA. So when the effect of that mutation is seen is when the result of the codon, in this case AUG, has now changed to AUA. And what that in turn translates to is that the amino acid which originally was meant to be methionine has now changed to an amino acid called isoleucine. And we refer to this again as a frameshift mutation because if we look at further down at the end of that sequence, we see that now there is a sequence that lacks a nucleotide. And this missing nucleotide has resulted in also an amino acid that is not produced in that sequence. So we have the effect of isoleucine being a new amino acid, but we also see that the chain lacks an amino acid. So the effect of that is significantly distinct. So insertion essentially refers to adding on or inserting. A gene mutation where one or more nitrogenous base is inserted or added on will result in a frameshift. Again, if we had a length of a gene and if we decide to add on into this, it's going to lengthen or increase the size of that gene. And so we refer to that having a shifting effect on the size of that gene. So after insertion, we find that all the base triplets are altered, and consequently, the amino acid sequence is changed. And if we were to look at that, we're seeing in this specific sequence, we're seeing that there is an insertion of a nucleotide here, and that insertion in turn has an effect on the mRNA sequence, and lysine has now been replaced with glutamic acid, and we see the effect of the entire chain of amino acids being altered. And often you end up having an odd nucleotide, and in this case as an amino acid plus an extra nucleotide that hasn't been coded for. And so when we look at this, this is the effect of insertion. So guys, we've looked at the effect of insertion deletion as frameshift mutations. We've looked at substitution as well as a point mutation. What's very important for us to look at next is the effect of these mutations on the protein and how that affect characteristics of individuals. You guys have done well. Let's have a short break and I'll see you in a bit.